Although we do our best to summarize veterinary articles, it does not supplement the actual article, and if you are able to download and review the article yourself, please see the link in the description. This will summarize the article titled, Creatine kinase and its isoenzymes in the serum and cerebrospinal fluid of healthy canines. Introduction. Creatine kinase CK is an enzyme involved in the transfer of phosphate from ATP to creatine, and it plays a crucial role in regulating the ADP-ADP ratio during energy supply disruptions. CK is found as three isoenzymes, CKMM, CKMB, and CKBB, which are separated electrophoretically and are tissue-specific in their distributions. In veterinary medicine, CK is commonly used as an indicator of acute muscle damage, and alterations in its isoenzyme distributions have been utilized as a diagnostic tool for myocardial infarction in humans. Recent studies have suggested that changes in serum CK isoenzymes are associated with neurologic disease in dogs, particularly elevations in serum CKBB and central neurologic disease. While serum CK has proven to be useful in diagnosing and predicting CNS disease in dogs, cerebrospinal fluid CSF-CK remains relatively unstudied in veterinary medicine. Studies in human medicine have shown that CSF-CK is useful in assessing cerebral infarct size, meningitis, head and spinal trauma, subarachnoid hemorrhage, and stroke. Analysis of changes in CSF-CK isoenzymes in response to neurologic disease has been scarcely studied in dogs. A recent retrospective analysis of 578 canine patients with various neurologic conditions found no significant correlation between CSF-CK levels and disease categories. However, no specific isoenzyme analysis was performed. Establishing reference intervals, RIs, for CSF total CK and its isoenzymes in clinically normal canines will enable a more thorough analysis of CSF-CK's diagnostic and prognostic roles in veterinary medicine. Thus, this study aimed to establish rigorous RIs for CSF total CK and its three predominant isoenzymes in healthy dogs. The study sought to investigate the distribution of CK isoenzymes in clinically normal canines, which to date has not been described. This study will enable a greater understanding of CSF-CK's potential diagnostic and prognostic value in veterinary medicine. In addition to measuring serum total CK and its isoenzymes, analyzing CSF total CK and its isoenzymes can provide more detailed information on the extent of neurologic injury and its prognosis in dogs. Material and methods. An observational study was conducted at the Washington State University's Veterinary Teaching Hospital, WSUVTH, to evaluate serum creatine kinase CK activity and isoenzyme distribution, as well as total CK activity within the cerebrospinal fluid CSF of clinically normal dogs undergoing general anesthesia for elective procedures. A minimum of 40 dogs per reference interval RI were recruited. All dogs underwent physical and neurological evaluations prior to enrollment, and only those with normal results entered the study. Venous blood and CSF samples were collected and evaluated for abnormal hematologic or cytologic findings. Dogs aged 2 months or older and undergoing anesthesia for spaying or neutering were eligible for inclusion in the study. The veterinary neurologist on staff conducted complete neurological evaluations prior to sample collection. Venous blood was collected from the jugular or cephalic vein and CSF via atlanto-occipital spinal tap. Serum CK isoenzyme analysis and serum total CK activity were performed on 100 microliter of serum. For CSF, 100 microliter was mixed with 1 microliter of CK activator and allowed to activate for 40 minutes, then ultrafiltrated in the 30 kDa Amicon filter. CSF total CK activity and CK isoenzyme analysis were then performed on the activated and ultrafiltered CSF. CSF samples were analyzed for cytologic abnormalities, nucleated cell count, and RBC count. Dogs with abnormal findings were excluded from the study. Relevant discriminant values are presented in Table 1. Grossly lipemic serum and serum with a hemolysis index value. 3 were excluded from serum RIs. Reference value advisor was used to calculate RIs consistent with the American Society of Veterinary Clinical Pathologists, ASVCP, and National Committee for Clinical Laboratory Standards guidelines, with each RI consisting of a minimum of 40 dogs. Box-Cox methods were used to transform the data where possible and create RIs with 90% confidence intervals using the robust method. Non-normal data that could not be transformed were analyzed using non-parametric methods, with RIs and 90% confidence intervals determined via the bootstrap method. Serum and CSF total CK activity was determined using the Siemens dimensions panned clinical chemistry system, with a within-lab coefficient of variation, CV for serum total CK of 2.8% and 2.1% for the low and high ends of normal range, respectively. The CV for CSF total CK activity was 7.2%. Serum and ultrafiltered CSF were electrophoresed using the Helena Laboratories Quick Gel CK Vis isoenzyme kit and Quick Gel chamber. QuickScan 2000 was used to determine CK isoenzyme distribution densitometrically. Prior to enrollment, written informed consent and health history were obtained from each dog's owner, and all activities were reviewed and approved by Washington State University's Institutional Animal Care and Use Committee. Results: a total of 55 healthy dogs were included in the serum total CKRI, and 45 of these dogs were used to establish the serum CK isoenzyme reference intervals. Similarly, 53 normal dogs were included in the CSF total CKRI, and the CSF CK isoenzyme RIs were established using 42 of them. Table 2, Table S1. 7 dogs were excluded from both the serum and CSF RIs due to CBC or serum biochemistry abnormalities, and 2 additional serum samples from healthy dogs were excluded from the serum total CK and serum CK isoenzyme RIs due to iatrogenic hemolysis greater than 3 on the HIL index. 10 dogs with insufficient serum and 11 dogs with limited CSF were excluded from the serum and CSF CK isoenzyme RIs, respectively. Thus, 33 dogs were used to establish reference intervals for all RIs serum total CK, serum CK isoenzymes, CSF total CK, CSF CK isoenzymes. Dixon's method detected one serum CK outlier, and this dog was excluded from the serum total CK and isoenzyme RIs. Additionally, the 2 youngest dogs had CSF total CK activities more than 2-fold higher than the highest value in the RI, resulting in exclusion from both the CSF total CK and isoenzyme RIs. 2 dogs had 1 or more CK-CSF isoenzyme activities that were detected as statistical outliers, and these were excluded from both the CSF total CK and CK isoenzyme RIs. The median serum total CK value for the included dogs was 159.0 units per liter, and CKBB contributed slightly more than CKMM. The reference intervals for serum total CK and serum CK isoenzymes are presented in tables 3 and 4, respectively. The median CSF total CK value was 3.7 units per liter, and CKMM was the predominant isoenzyme in the CSF. Table 4. Discussion. Currently, there are few studies reporting reference intervals, RIs, for cerebrospinal fluid CSF creatine kinase CK and its isoenzymes in dogs. Although some studies have presented different CK isoenzyme activities, Table S2, variations in sample handling, storage, and isoenzyme detection techniques may contribute to the differences noted. The stability of CKMM, CKMB, and CKBB varies at different storage temperatures and lengths, which may account for some degree of the variability noted. Our study attempted to reduce this factor by running all samples within 8H of collection. There can be significant inter- and intra-individual variation in total serum CK and CK isoenzymes, depending on the conditions under which the sample has been obtained. Our study made every effort to reduce these factors of variability, but this may nonetheless contribute to the wide serum CK isoenzyme RIs established in our study. The method of detection of CK isoenzymes may also affect the results obtained. It is possible that studies describing CK isoenzyme distributions, fractionated via Western blotting, have reported incorrect estimations of the actual CK isoenzyme distribution. It is important to note that our study's findings of CKMM predominance in canine CSF are different from findings in human CSF and canine brain tissue homogenates. While CKBB is predominant in humans, CKMM is presumed to be due to blood contamination since the distribution of CK isoenzymes in the CSF of clinically normal human patients is not available. Although the presence of CKMM in our canine CSF samples may represent a low level of contamination from penetrating muscle layers in the dog's CSF tap, it is important to note that CKMM has also been detected in the normal CSF of other species such as rabbits. Our finding of CKMM predominance in normal canine CSF may be especially useful as elevations in CSF-CKBB have been demonstrated in cases of neurological disease. Therefore, our finding suggests that any detectable elevation of CKBB and canine CSF may be a marker of neurological disease. A limitation of this study is the low sample size. However, due to the risks associated with the collection of CSF, a more conservative number of animals based on published guidelines were deemed acceptable for the establishment of these RIs. This study is the first to describe the normal CSF-CK isoenzyme distribution in healthy dogs, establishing normal RIs for CSF-CK and its isoenzymes in dogs is a critical step in determining the clinical usefulness of CK and its isoenzymes in disease.